Hello, and thank you for joining the presentation. My name is Michele Korfin, and I just joined Gamida Cell a few weeks ago. I've been very honored throughout my career to have the opportunity to work on many oncology therapies that have had significant impact for patients and their outcomes. And what brought me to Gamida was that opportunity to work on a therapy such as Gamida Cell that will be evaluated or was evaluated in clinical trials for patients with a great unmet medical need, and also the excitement of our NK platform, which I will also talk about during this presentation. So I'm very excited to be part of the Gamida team and to share our presentation with you today. Some of the statements I'll make during this presentation may contain forward-looking statements. I refer you to our disclaimer here on this slide and also in our most recent SEC filings. So at Gamida Cell, we are developing advanced cell therapies. The first investigational therapy that I'll speak about is Gamida Cell. So Gamida Cell has FDA breakthrough designation. Gamida Cell was studied in phase 1/2 and most recently in a phase 3 trial for patients with high-risk hematologic malignancies in need of a stem cell transplant. We presented our top-line data in the second quarter of this year, and we anticipate full data presentation in fourth quarter and also to start the BLA rolling submission in fourth quarter. Gamida Cell is also being studied in patients with severe aplastic anemia in the phase 1 setting at this point in time, and we're excited to continue advancing that clinical data set. During this presentation, I will also discuss GDA 201, which is our NK investigational therapy or natural killer cell investigational therapy. We have an ongoing phase 1 study in non-Hodgkin lymphoma and multiple myeloma, and I'll be able to provide some updated data for the non-Hodgkin lymphoma patients. Both Gamida Cell and GDA 201 are part of our proprietary Nand Platform technology. The Nand Platform technology focuses on enhancing the number of allogeneic donor cells and has the potential to expand any cell type. In addition, the Nand technology does preserve cellular functionality and phenotype. So I will start with our investigational therapy, Gamida Cell. I want to introduce you first to Stacy. So Stacy is a patient from our first clinical study of Gamida Cell at Duke University. Stacy was diagnosed with AML. Unfortunately did not have a matched related donor, and really unfortunately at that point in time had limited treatment options and was able to enroll in our clinical study. I am so excited for Stacy and for her family that she has now been in remission since her bone marrow transplant, which was approximately 9 years ago. We thank Stacy for her participation in the study, and we thank all the other patients who've participated in both our phase 1/2 and our phase 3 study. And thanks Stacy for allowing us to share her story. In the United States, there's approximately 120,000 patients with hematologic malignancy who are looking to actively seek a treatment option each year. Of those patients, you see about 13,000 patients who are eligible each year for transplant. There's a great unmet need still in transplant for these patients. So let me start from left to right in this depiction. Over 5,000 patients are eligible for a transplant, but unfortunately do not have a match. And in many cases, these patients are not even referred to a transplant center. When we talk about Gamida Cell and our future potential, that is an opportunity to expand the market, because unfortunately for these patients, they've run out of treatment options and they're not even being referred to a transplant. The next group of patients are the over 5,000, are also approximately 5,200 who do not have a matched related donor. These patients are undergoing different types of transplants, whether it's haploidentical or matched unrelated or cord blood graft. And as you see from the published literature, the outcomes are not always as ideal for these patients. So this is an opportunity where in the future, upon a potential FDA approval, Gamida Cell could help to improve the outcomes of those patients. And then the third group that I'll talk about is those patients who are fortunate to have a matched related donor. In some situations, though, unfortunately, the donor is not able to make it to the transplant center, or the patient doesn't have timely access to the donor. So there is still some potential opportunities that are unmet for those patients. I want to talk about the importance of age for donor source. This is a very important finding that has been published in the past. And just today is the Cord Blood Connect meeting has started virtually on September 10th. And we had an ongoing collaboration with CIBMTR to explore outcomes, contemporaneous data, looking at donor age and the impact on survival. And what you'll see on the Kaplan-Meier to the right of this slide is the fact that there is a statistically significant difference based on donor age. So if a donor is less than or equal to 30 years, the recipient has a statistically significant greater survival probability. And if a donor is greater than 30, the outcomes are not as great. So we looked at stem cell transplants across 3 graft types, both matched unrelated, mismatched unrelated, and haploidentical donors. So this is a very important finding. And when we're talking about Amadubicell's clinical data and the potential, Amadubicell starts with stem cells. So that's deemed to be the youngest cell type used in allogeneic bone marrow transplant. So that correlates in an important fashion with this takeaway around the importance of donor age. Let me now talk about the clinical trial and the clinical data for Amadubicell. So this is our phase 3 study. This was a global randomized study conducted at over 50 sites. Inclusion criteria allowed for patients aged 12 to 65. Patients had to have high-risk hematologic malignancies and deemed to be eligible for allogeneic bone marrow transplant, and no matched donor available to the patient. Patients were randomized 1-to-1, either Amadubicell or the comparator arm, which in this case was standard cord blood. Primary endpoint, time to neutrophil engraftment, which we'll talk about the clinical meaningful nature of that endpoint as we present the data. Secondary endpoints and other additional endpoints included platelet engraftment, infection, hospitalizations, GVHD, certainly adverse events, and non-relapse mortality and disease-free survival. I'm able to present our phase 3 primary endpoint at this time, which was a very compelling, encouraging result. So Amadubicell significantly reduced the time to neutrophil engraftment. As noted in the table, Amadubicell's median time to neutrophil engraftment was 12 days as compared to the comparator arm or the control arm, which was 22 days. And this was highly statistically significant. The demographics and baseline characteristics were well balanced in the 2 arms, and Amadubicell was generally well tolerated. We will be presenting the secondary and additional endpoints later on this calendar year, and certainly presenting additional safety data too. Engraftment has been demonstrated in the past to be a key milestone in recovery, and rapid engraftment is also associated with fewer infections and shorter hospitalizations. Shorter hospitalizations are important, obviously, from the patient standpoint, because the patient's able to get out of the hospital sooner, be with his or her family, be back home, but also from the overall system standpoint, less time in the system is recognized to be more beneficial for the hospital, but also from the payer perspective. I also want to talk about the cumulative incidence of neutrophil engraftment, and in the as-treated population, as depicted in this figure, you see that Amadubicell had a 96% cumulative incidence of neutrophil engraftment as compared to the control arm, which was 88%. When you look back to our phase 1-2 data, we did demonstrate the correlation between rapid engraftment and fewer infections and shorter hospitalizations. So from the standpoint of infections, both grade 2-3 total infections and overall bacterial infections, you saw a statistically significant reduction in the Amadubicell arm as compared to historical data for standard cord. And also in the case of hospitalizations, you saw a statistically significant reduction in hospitalizations for Amadubicell. So this was from our phase 1-2 data, and we look forward to presenting the secondary and additional endpoints for our phase 3 data later this year. Now that we have completed our phase 3 study and read out the primary endpoint and soon to read out the additional endpoints, we at Gamida Cell are now preparing for a successful Amadubicell launch. The absolute focus of this launch has to be assuring a positive patient experience. These patients have a great unmet need. They've been through so much in regards to their diagnosis and prior treatments. Our focus and responsibility will be assuring a positive patient experience. 4 key aspects of our launch: educating the top transplant centers of our clinical data, working with payers to ensure reimbursement. We will and are building an important patient and hospital support service, and also making sure our manufacturing will be ready for commercial launch. So we're confident in terms of our launch preparation for Gamida. And just to summarize, as I transition to our additional pipeline, Gamida does have the potential to be the first FDA-approved bone marrow transplant graft. We've had very encouraging, compelling clinical data so far, unprecedented time to neutrophil engraftment, and the side effect profile was deemed to be well tolerated. In our earlier data set, we did demonstrate a reduction in hospitalization time and decreased risk of infection. We will be initiating our rolling BLA submission in fourth quarter of this year, and then beginning our commercial activities to be ready for a potential second half of 21 launch. Let me now transition to GDA201. This is our natural killer cell investigational therapy. I want to introduce you to Wayne. So Wayne participated in our phase 1/2 clinical trial at the University of Minnesota. Wayne had been diagnosed with lymphoma and was heavily pretreated before he entered our study. Our study represented Wayne's 5th line of therapy. Prior therapies were deemed to be relatively aggressive. We are proud to say that Wayne remains in remission following his treatment with GDA201, and we thank Wayne and the other patients that participated in this important early phase 1 study for us. So the phase 1 study of GDA201, we included patients with both non-Hodgkin lymphoma and multiple myeloma. We'll just be discussing the lymphoma data, which is what we have to present today. Patients received fludarabine cyclophosphamide for a lymphodepleting regimen, and then they received GDA201 in combination with either Rituxan if they were a lymphoma patient or Implicity if they were a myeloma patient. Given this was a phase 1 study, the primary endpoint was the maximum tolerated dose of GDA201, and 3 doses were evaluated. Secondary endpoints were overall response and toxicity. Now, even though the primary endpoint was focused on dose, we were very encouraged to see the efficacy of the lymphoma patients that we're presenting here. So if you look at the swimmer's plot, you'll see that 7 of the 11 patients had complete responses. These were durable responses, as you see depicted in some of the patients, across both follicular lymphoma and diffuse large B-cell lymphoma. So we're very encouraged by this efficacy from the early study. In terms of key takeaways for GDA201, really 2 key points. Promising early clinical activity, 7 complete responses and 1 partial response among 11 heavily pretreated patients with lymphoma. Activity observed not only in follicular lymphoma, but also diffuse large B-cell lymphoma. And the maximum target dose was achieved. The side effect profile was deemed to be generally well tolerated. So no dose-limiting toxicities or DLTs, no graft versus host disease was observed. And also with some of the other new therapies that are being evaluated in lymphoma, you are sometimes seeing neurotoxicity or tumor lysis syndrome. Neither of those were observed in our study. So we are excited now to advance the clinical data plan for GDA201. In terms of next steps, we will complete the readout of this phase 1 study. We're also working with our manufacturing and research colleagues to finalize a cryopreserved formulation, which will allow for broader use in both clinical trial settings, but also potentially for preparing for future commercialization. And we are looking to file the IND in the first half of 21. Future work that we'll also consider is combining with a range of antibodies, and also potential evaluation in solid tumors. And in addition, potential genetic modification of the NAM expanded NK cells. So to summarize at Gamida, we are very excited by the clinical data that we have seen so far for our Nand platform, which has the potential to expand any cell type, including stem cells and NK cells. For Gamida, a very exciting time for us as we look to read out the additional endpoints later this year, and also to prepare for the BLA submission and potential launch. GDA201, promising early clinical activity in patients who are heavily pretreated with lymphoma. And we have the opportunity to potentially launch the first ever FDA-approved bone marrow transplant graft. I am so excited to be part of the Gamida team and was excited to share this very encouraging data with all of you.